Thank you and good morning. Nice to see you all here, and nice to talk about Kyverna. We are also in the autoimmune disease. We are applying cell therapies to the treatment of autoimmune disease. There are over 80 known autoimmune diseases. There are suboptimal treatments, and there are no cures. And our vision is to build the industry leader in engineering and developing a new class of curative living medicines to treat these diseases. We would do this through a deep understanding of the disease itself, and then select the ideal approach to treat the diseases, because one approach does not fit all, particularly with the diversity of diseases that are there. We also have a very good team of leaders. We have experts in autoimmunity, and we have experts in cell therapy. Dominic Bory is our CEO. James Chung is our Chief Medical Officer. I'm the Chief Technology Officer. And Tom Van Blarcom comes from Allogene. He is our Head of Research. We bring a deep understanding of both the disease and its drivers, as well as ways to approach those diseases and bring true benefit to patients. We want to be the first mover, and we have a lead asset, which is an anti-CD19 CAR, to be viewed in the diseases where B cells contribute to the pathogenicity. Our second approach is through regulatory T cells. By synthesizing regulatory T cells to calm the immune system, we hope to treat other diseases that do not have B cells as a part of the pathogenicity and bring benefit, curable benefit to patients. This is our pipeline. Our lead asset, KYV-101, is entering the clinic at the end of this year. We have an allogeneic platform that we will put the same lead candidate onto and look at providing an allogeneic option to these patients. And then we have a couple of different programs that are in early discovery on the regulatory T-cell pathway. To talk about KYV-101, we know, you've also just heard also, that B cells do create to the pathogenesis of this, or contribute to the pathogenesis of this disease, and tissue-resident B cells are very difficult for traditional therapies to access. So with a cell therapy approach, we can access those tissue-resident B cells and provide ablation that will then bring the score on the diseases down to 0. There is transformative efficacy, as has been highlighted in the Nature paper from Dr. Shett. And a lasting immune reset may be possible. We're still looking into that. There was complete withdrawal of background immunosuppressants, and it avoids dire consequences of long immunosuppression. We have a fully human FCFV, and we have a CD8 hinge and a CD28 transmembrane and co-stimulatory domain. We believe that this construct is going to be very valuable in an autoimmune setting, given the fact that it is fully human and that the co-stimulatory domain, whoops, went too far, is engineered to provide lower levels of cytokine secretion. We licensed this construct from the NIH. It was looked at in 20 patients with lymphoma, and it did show improved safety, lower cytokine release and ICANS, and lower risk of cytokine-driven flares. Moving on to the allogeneic platform, we are partnering with Intellia and their technology, which provides good protection of the allogeneic cells from the host. In their approach, they avoid the rejection through CD8 cells, also through CD4 cells, also through NK cells, and they avoid deep immunosuppressive therapy in order to gain engraftment. So we think the combination of our construct on the allogeneic platform provides real options for patients with autoimmune disease who are very different from patients with cancer. And we're hoping that the characteristics of our construct prove out when we start a clinical study at the end of the year. Our other approach is through synthesizing regulatory T cells, and their mechanism of action is not an effector mechanism, it's a suppressive mechanism. Here we engineer FOXP3 into normal T cells, and then we use targeting through CAR Ts to find the site of inflammation and provide localized suppression of the immune response. By using this approach, we have a higher abundance of T cells that we can use therapeutically in patients. Our proprietary regulatory T cell program avoids complexity. It leverages the standard CAR T approach, which gives us a very well-defined product and one that we can track through patients. We believe that this approach also significantly reduces costs and improves access to patients. We are navigating what is next. We are building ourselves to be the industry leader in cell therapies that address autoimmune disease. We have a good set of collaborative partners, and we are using the science and innovation that is evolving every day to advance our programs and our knowledge in this area and bring real health to patients who don't have real options. Thank you.